Hey everyone, so welcome back. This week we're going to talk about phylogenies, and I'm going to talk about some basic aspects of phylogenies, and then Laura is going to talk about the building. And the first thing I think is important to start out with is that phylogenetics is really important. So we're going to talk about phylogenetics focusing on evolutionary and ecological questions, but phylogenetics is really important for medical type questions as well. So for example, the coronavirus situation that we're dealing with. If you take the coronavirus sequences from patients, such as they did in this tree, and then compare it to coronavirus sequences from other animals or other types of coronavirus, and place them all in a phylogenetic tree, you can figure out where our coronavirus originated from. So if you look at this tree, where they've sequenced a bunch of coronaviruses, you can see that the human patients with coronavirus fall very close to the bats with this coronavirus strain. You can see that they share a most recent common ancestor that's before any other types of animals are in this tree. And this allows us to infer that the coronavirus we are dealing with right now originated from bats. But we're going to focus on a different type of phylogenetic tree. We're going to focus on an organism-based phylogeny, and these phylogenies were interested in features that organisms have. And to talk about features, it's really important to talk about how features can evolve. So one mechanism for how features can evolve is through homology. So homologous features are features where you have a shared ancestor. And these features, when we talk about them, we could talk about them in terms of genes or proteins, but we can also talk about them in terms of structures. And we're going to focus on structures just because it's easiest for us to deal with. And one thing that's really important when you're talking about homology is that homologous features can appear both similar to one another and different. So my favorite example of homologous features is in the fin to limb transition with humans. So as humans, we have these wonderful arms and legs, and our arms are actually homologous to the fins of a fish, specific fins, but that's because we have evolved from a fish, and so you can refer to our arms and our legs as being homologous to certain fins on fish. So in that case, these structures would appear different. However, you can also say that our arms are homologous to the arms on a gorilla or a monkey or an alligator or a frog, and in those cases, those arm structures look very similar to one another. And so that's just an important aspect of homology to keep in mind. Another type of way that features can arise is through convergent evolution. So these are analogous features where the features appear very similar to one another, but there's no common origin. And in this case, the organisms have developed the same features because it is beneficial to them. So my favorite example of this is some fish I used to study called electric fish. Electric fish are incredible because these are groupings of fish that have evolved electric organs, and there is no common ancestor to many of these lineages where the common ancestor had an electric organ. So if we look at this phylogeny to the left, we can see that there are a number of species have electric organs, and so those are these little drawings here. However, if we look at these little red ticks, which is the lineages that have evolved them, we can see that there are not common ancestors for any of these lineages that have electric. So this means that this trait has independently evolved at least 6 times in the history of fish, which is incredible in my mind. But this can cause a really big issue when we are putting these fish on a tree, right? Because if we are just looking at the trait of an electric organ, we may accidentally put all of these fish on the same branch when actually they're from completely different lineages. So this highlights one thing that's really important when we're making our phylogenetic trees is you have to tease out these features that have arisen through convergence from other features. So when we're doing this, we're going to look at some phylogenetic trees, and we're going to try and make them, but this is something we will talk about a little bit more. So when we are interpreting phylogenetic trees, the first thing that's really important to notice is this thing at A. So this is our most recent common ancestor of all of the species on our tree, including this guy down here, our gar, and all of these lineages here. What we see at A and what we see at B are called nodes. Nodes show where two lineages have diverged from one another. And then after a node, we see a branch, and this shows our lineage that has diverged after this node. And the one thing that's important is that I'm using the word lineage here. So lineage can describe more than one species. I am using the word lineage here because say for one, there are more than one type of seahorses, but also if we look at where the northern pike has diverged from these guys, we can see that there is a lineage here that's leading up to the divergence between seahorses and Medaka, and I just don't know exactly which species has been involved with that transition. So we call it a lineage just to be a little bit more broad. And then when we're also looking at a phylogenetic tree, it's important to note that there are different ways we can classify organisms within this tree. So for example, sister taxa are species or taxa that have diverged from the same most recent common ancestor, and this happened at the closest node to them. So for example, seahorses and Medaka are called sister taxa in my phylogeny because they both diverged most recently from this bee. However, seahorse is not a sister taxa to the northern pike because the seahorse has not most recently diverged from this node in particular. It's most recently diverged from this node. In addition, another important way to look at the organisms on this tree is by using an outgroup. Outgroups are really great because they allow you to assess the ancestral state of all the organisms on your tree. Outgroups are species that are outside of your grouping of interest. So my tree is looking at teleost fishes, which is not important for you guys to know, but the reason why I use gar as my outgroup is because gar are not teleost. So you can use any species. It's good to use a species that is very recent from your grouping of interest because they'll have more of the ancestral traits that you're interested in. So if we were looking at, for example, the mammalian lineage, it might not make a lot of sense to use gar as our outgroup. It would probably make more sense to use, say, frogs that are a little bit closer to us. And then also when we're talking about phylogenetic trees, it's important to talk about traits once more. So features are often used in these phylogenetic trees for organisms to denote where different organisms have diverged from one another. So features can be shared where two lineages have that same trait in common. So for example, ray-finned fishes and tetrapods both have bony skeletons. Traits can be derived. In a derived trait, those are traits that are new for a particular lineage. So for example, the amniotic egg is a derived trait for this lineage here that includes mammalian organisms as well as reptilian organisms. Furthermore, traits could also be ancestral. Ancestral traits are traits that the ancestor of that organism already had. For example, if we look back at our mammalian and our reptilian lineages, being tetrapods is an ancestral state because the amphibians out here already had 4 limbs. And it's also important to note that a trait could be shared and derived, or it could be shared and ancestral. It cannot be derived in ancestral though, so that is important to know. But when you're building your phylogenetic trees, you have to keep a couple of things in mind. So the biggest thing to keep in mind is we're going to be building our phylogenetic tree from synapomorphies. Synapomorphies are shared derived traits. So these are traits that we talked about, such as the amniotic egg. it's shared between all of the organisms on this branch, and it's derived where these organisms are the only ones that have it. And so we want to use synapomorphies to influence the trees you're making. But as we talked about earlier, there are some ways that you can get kind of tripped up when you're making your phylogenetic tree, and these are through homoplasies. Homoplasies include convergence. As I talked about earlier, you can have two organisms that are distantly or that are not related having the same trait. So like my electric organs. That is an example of convergence. They do not have a shared ancestor with electric organs, but they both have electric organs. Putting them on the same branch would be incorrect. And so you really need to watch out for traits that are the result of convergence. In addition, there can be reversals. And reversals is when an organism has derived from a common ancestor that has a specific derived trait. However, the organism itself might have the ancestral trait because it reversed back to that ancestral trait. So it's important to keep that, that in mind as well, because that is not an example of that organism having that ancestral trait to begin with. So it's different. And what you want to do in these cases is just rely on your outgroups with the ancestral conditions to make your tree. That was going to be the biggest help for you. And then just looking at all of the traits that are provided. But Laurel's going to talk about that a lot more. So watch Laurel's example video and enjoy your lab.